Hello, my name is Shada Aquino, and I'll be presenting on the effects of Toxoplasma gondii infection on amyloid plaque accumulation and microglia activation in the Alzheimer's disease. So to begin with a little bit of background, Alzheimer's disease is a form of dementia that is typically characterized by the accumulation of beta amyloid plaques and neurofibrillary tangles in the brain. The Arctic mouse model being used in my project produces these plaques at about 2 to 4 months of age, which we see in this graph over here. The parasite that we're looking at is the Toxoplasma gondii, which is a parasite that can infect most warm-blooded animals and has been found to play a role in altering immune activation. We are also looking at the C5a receptor 1, which binds to C5a, a pro-inflammatory complement factor, and this also plays a role in immune response to infections. So in terms of the rationale behind this project, the Ladone lab, which is at UCI and also a collaborative lab with ours, observed reduced plaque pathology and an increase in microglial macrophage presence in 5X FAD Alzheimer's disease mouse models 6 weeks after infection with this parasite. And other literature with the TG2576 and J20 Alzheimer's disease model have found the same as well. So we are trying to determine if these changes also occur in the more aggressive Arctic mouse model, determine if C5AR1 contributes to any changes in pathology, and if these changes are maintained 12 weeks after infection. In terms of our methods, we had Arctic and Arctic C5AR1 knockout mice that were injected with either PBS as a control or about 200 parasites 6 or 12 weeks before their perfusion date at 24 weeks of age. Their half-brains were dissectioned, performed immunohistochemistry, and we took slide scanner images to be processed on Imaris for analysis. So in terms of our results, this is from the 6 weeks post-infection group. As we see over here, thioflavin stains for amyloid plaques, and PBS, which was our control group, had a great number of plaques in the hippocampus region, while those that were infected with toxo had a greatly reduced number of plaques in the region. And this was true for both the Arctic and the Arctic C5AR1 knockout mice, as we see in these graphs over here. On top of that, we had the IBO1, which stands for activated microglia or peripheral monocytes, and we see that there was greater activation in the Toxoplasma infected groups. And we see that in this graph over here as well. Meanwhile, GFAP, which stands for astrocytes, seemed to have no changes between the treatment groups. Moving on to 12 weeks post-infection, as we see in all these images here, it seemed the IBO1 staining as well as the thioflavin staining returned to uninfected levels, and that was also represented by the graphs on the side here. So as for a quick summary and conclusions, injection with the parasite does lead to a reduction in amyloid plaque load in the hippocampal region of Arctic mice, which does corroborate other data. The Toxoplasma gondii infection recruits more microglia or peripheral monocytes to the hippocampus region, which could explain the reduced amyloid plaque load. However, the effects seem to be mitigated by 12 weeks post-infection. There were also no differences seen in GFAP staining or the presence of C5AR1. As for future direction, we will be performing more immunohistochemistry, more particularly in C5AR1, IBO1, and amyloid, as well as C1Q, C3, and GFAP. And that is all. Thank you.